Hello everyone. My name is Dr. Erik Karlsson. I am the Deputy Head of Virology at Institut Pasteur Cambodge. I have a number of other titles, but I want to thank, first of all, obviously, Dimitri and everyone at IP Korea for having me here to talk today. I'm sorry I cannot be in the room with you. I really was trying to, but things have been a bit complicated with my travel schedule, but I'm still glad I get to talk to you. So I'm going to talk to you a little bit about what we're doing at IPC along with our partners on leveraging sort of novel technologies and sequencing for surveillance at high-risk animal-human interfaces. So just for those of you that don't know, I'm sure that many of you are aware, but obviously Cambodia is a small country in the Indochina Peninsula. It's a least developed country with a population of about 17, 18 million. It is a hotspot for emerging and endemic infectious diseases. There's a number of issues in Cambodia, and there's a large dependence on agriculture. So, about 60% of all Cambodian households have some sort of agricultural holding, and the majority of those actually raise poultry. So there's a very high animal-human interface in Cambodia. There's also a lot of tourism. So we have a very large dependence of our GDP on tourism. So there's a lot of people coming in and out of the country at all times. And those people are interacting not only with the population, but also with wild animals, with domestic animals and things like that as well. So it's something we have to keep an eye on. In terms of Institut Pasteur Cambodge, in case you didn't know, we've been a non-for-profit research institute of public utility. So we are actually a public utility under the Ministry of Health since 1953. And so we have a number of different mandates, including research as well as training and then diagnostics. So we've been, of course, studying a lot of different human viruses. I'm coming from a respiratory virus background, so obviously we've been studying a lot of human seasonal influenza as the NIC and the reference laboratory. We study a lot of other respiratory viruses, including SARS-CoV-2, but also things like HIV, enteric viruses, and many, many others. And that's just the virology portion of what we do. But you have to also remember that we've spent 25 years of surveillance at the animal-human interface in this country. And so we've been focusing on not only humans, but also things like poultry, as well as swine, and then, of course, even things like rodents, and of course, everyone's favorite, bats, right? So lots and lots of different animals that we've been looking for, lots of different viruses, including vectors, hello, Anavaj, good to see you. I know we've worked with Anavaj quite a bit in the past, and many of you as well on vector and vector-borne disease as well. And so we are leveraging our capacity, especially capacity that we've been built up over COVID-19, not only to improve our capacity and capability for surveillance of these pathogens, but also for sequencing. So we've developed our sequencing platforms up quite a bit, and we're sequencing all of these viruses that you see on this screen at the current time, as well as many others, often in real time as they occur in our surveillance programs. Because surveillance and genomic surveillance is so important, we've been incorporating new techniques. So one of them is this rapid avian influenza surveillance that we've been doing using what we call an integrated indexing and field-forward bioinformatics. Basically, we have been working with our collaborators at Johns Hopkins University, which uses an indexed primer set that allows us to universally amplify all genome segments of avian influenza in a very short amount of time and skip a number of steps within the sequencing process. At this point, we can basically go from sample to sequence in less than 7 hours, which has been absolutely critical, not only for things like surveillance, but also responding to human infections. So we've used this quite usefully in 2 H9N2 infections, one in 2021 and one in 22. And then, of course, this year we've had now, since last week, 6 human cases of H5N1, Here's just an example of how we've used this in February of this year, where we had 2 cases of avian influenza infection. Everyone was quite worried it would be 2344B. The ability to go from direct sample to sequence in less than 24 hours allowed us to determine this was 2321C, similar to what had been circulating in the country at the time. It also allowed us to genomically link case 1 and case 2 together and understand that they were exposed to the same source and that there's no evidence of human-to-human transmission at the current time. So these have been really small advancements, but they're absolutely critical not only for surveillance, but also response in real time. Lately, what we've been doing is looking if we can expand our surveillance program. So instead of going out and sampling a bunch of animals and a bunch of humans, we're really looking at, much like the rest of the world, how environmental sampling can actually improve our surveillance prognostics, as well as coupling that with novel diagnostics for early warning. So I think, as many of you know, early warning is not easy. It's not very clear all the time, but it's important that we have a rapid response, not only to respond to an outbreak, but also to detect and prevent those infections and anticipate those infections in real time as well. That kind of early warning requires active surveillance. And so for anyone that's done active surveillance, we know it's costly and it's time consuming. It requires a lot of effort by a lot of people, and it's often not done in the easiest of places. So you see here my team out in a boat in the middle of a flooded lake going out to a wild bird colony. So, you know, these things are not always done in our backyards. We have to travel very far, and it's not easy. And we have issues with sample shipment, et cetera, et cetera. Environmental samples really present a viable option to improve early warning because if we use them with in-field diagnostics and discovery, it can not only increase our surveillance capacity, but it also contributes our understanding to new and emerging pathogens and high-risk interfaces. It increases our speed of detection, our comprehensiveness. So we're looking for in a pathogen agnostic way. It increases the biosafety of our teams and the people that are involved with this kind of work because they're not actually directly involved with the interfaces at some points. It also increases animal welfare. So I think sometimes when people come from the public health field, they forget the animal health aspects of all this, and that handling these animals, going in and stressing them out, actually decreases animal welfare. So the ability to take environmental samples does both for both humans and animals. And then, of course, cost. If we can take fewer samples, get more information, and be able to use that for direct response and action, that reduces the cost of our surveillance in that particular spot. And we can then expand that surveillance out and do more potentially with less funds. So we've been doing environmental surveillance in places like live bird markets. So this is a live bird market in Phnom Penh, using a number of different sample types. So here you see what kinds of things we've used in the past. A lot of other groups use these all over the world. But mostly what we've been focused on since the pandemic and recent times is air. And so what we've been doing is putting air samplers in these markets and comparing those directly against our gold standard, sort of take it directly from a bird, PCR techniques and things like that. So here you see an air sampler in a market next to some holding of birds. We've been putting it in a number of different markets and a number of different contexts. Here you see one in more of an open air market next to the slaughtering point in that market. And what we found is that we can actually use these air samples to actually detect AIV. And so I'm using avian influenza here as an example because we have the most data on it, and it's easy for me to convey what we're seeing. So if we look at viral load in these samples, of course, in the birds, there's going to be the highest viral load, but we can actually detect these viruses not only in slaughter, but also in storage, which is separated from slaughter, and then even in food sales, which is physically separated from these other two places. So we're actually able to detect a gradient of these viruses over the market. And we've been able to actually correlate this with detection in the markets where we're looking at our total sampling periods, and we're able to detect avian influenza in the air over approximately 50% of the time that we're there detecting it, many of these at the peak of AIV detection in birds in the markets as well. We are detecting multiple subtypes in the air. So as you can see here in all of our samples, we've detected that are flu positive. We've detected H9, but we've also been able to detect H7 and H5. And these are in multiplex combinations where in some of the samples, we're detecting H9, H7, and H5 in the same exact sample that we're detecting in. So that indicates a huge diversity of these viruses in the air of the markets, which we may not have picked up by just sampling individual birds. In addition, we're using different types of samplers. So here you see a BASH sampler on a bird cellar. We've had experiments where we've had them wear these samplers for just 30 minutes a day for 1 week during either peak or low season for AIV. And what we find is there's 100% chance of an individual being exposed to a viable, so we can actually culture out of these, a viable H5N1 or H9N2 virus in just 30 minutes during peak AIV periods. So you can imagine how much that changes our understanding of risk and our need for interventions in these markets as well. We've also been using this in a number of different contexts. So here you see a bat farm in Cambodia. So they actually farm bats for guano. These palm fronds are actually filled with these colonies of 30 to 50 Scotophilus bats, which are insectivorous. So here you see the colonies on the right. So it's kind of a zoomed in video from those palm fronds. And so we've been doing studies where in the past we were looking at feces and soil and urine from these animals, but also now we're looking at air. And so we've been able to put air samplers in these farms as we're sampling them over time. We're also looking at things like fruit bat roosts. So here you see my team sampling from a fruit bat roost in Cambodia with a number of different samplers underneath. And then, of course, bringing them into caves where possible and actually sampling the air inside caves as we're taking samples as well. We also use these in other places like swine abattoirs. Again, in the past, we would be using sort of what I consider the normal environmental samples, but now we can also use air as one of the ways to actually look at the overall composition of what's going on in these abattoirs, especially overnight where they have large number of pigs coming in from different farms and then being slaughtered on site. We also can now do this handheld. So we have these small biochemical collectors that are about the size of a pack of cards. What you can see here is one of these collectors in a hand that the air is actually going in through that small green chip in the top of the collector. It is electrochemical, so it pulls the air over, removes the nucleic acid, and spits out everything else. So the sample is completely inactivated, but we can also take that directly off of that chip and run PCR. We do not have to do an extraction step with these samples, which immensely increases our time to detection. So we've been using these in a number of different high-risk interfaces. So here you see a defeathering machine. And I get questions all the time whether or not we can actually use these to detect avian influenza. And indeed, we've used them in a number of different contexts. And we are able to detect avian influenza in air, especially in the highest risk areas, which are places like slaughter. And these studies are ongoing in terms of time and validation and things like that overall. What's great about these is we can use them for a number of different contexts. So we can get right up close and personal with things like coughing pigs, et cetera, and get individual samples from these animals over time and actually see what they're spreading throughout the populations. We can bring them with us when we're sampling in the wildlife value chain. So here you see my team sampling civets that are captive in a pagoda. And then, of course, for things like bat caves where we're looking at millions and millions of bats at one time. They're either flying over these large samplers, or we can use them individually in caught nets or during slaughter, not slaughter, but during sampling to observe what may be in the air or what the bats are actually putting out into the air individually. The advantage of these as well is that we can actually go remote. So we have the bright idea to strap them to things like drones. They fit on a drone quite nicely. So you can see here one just literally taped to a drone, but we can also do things like go buy remote controlled cars from the toy store and make what we call terrestrial drones, which we can use to drive into places like caves and farms. And we have been using these in the field. So here you see a drone in a bat cave actually sampling air from that cave. And you can see all the particulate that it's kicking up and actually pulling into that air sampler. And then, of course, driving the cars into places like swine farms, which is critical to be able to assess multiple farms in one day, also to prevent any kind of transfer of any pathogens from one farm to another, where we don't have to go home, take a shower, change all our PPE. We can just disinfect the terrestrial drone and take it to a new location, and it's not invasive on the animals. We also use many other kinds of sample types, things like ropes and wash water, wastewater, et cetera, to be able to collect these samples and really get a good comprehensive picture of what's going on. We also use it in the wild. So we use the drones in the wild to observe things like poultry or outbreaks in wild birds. So on the left, you see actually a video of a colony of wild birds in Cambodia. All those little white spots are actually birds, and we're looking for dead birds within that colony. We can then immediately go to that location for those dead birds and use our samplers in that context to get samples from those birds. You don't have to handle the carcasses or take them back for necropsy. And we can identify things like unknown duck farms or poultry farms nearby those colonies, as you see on the right, where we're actually able to go sample and look for any spread between the wild birds and poultry in real time. So this has worked very well for outbreak response in the past. Now, moving further, we're trying to combine environmental sampling with metagenomics. And so this is kind of, the next step in all this is, can we actually move this even further and make a pathogen agnostic approach, take an environmental sample, then couple that with metagenomic analysis to get a really good idea of what's going on. And so, this gives us a number of advantages. Obviously, being unbiased, we can do discovery, it's broad and dynamic. And then, of course, we can also get functional information about things like vaccine escape and antiviral resistance and other things from this data that we potentially couldn't get just from a PCR reaction for one virus alone. And so we've been doing this with a number of different samples. And so just as an example here, if I just take poultry samples and do metagenomics on those, obviously I'm going to get mostly influenza sequence because those samples were influenza positive. You can see here that 97% of that sample was actually influenza. But if I just take one single air sample taken at the same time, I still get that full genome of influenza at about 14% of the sample, but I'm picking up all of these other viruses, many of them full or almost full complete genomes, and actually able to understand what's going on in that environment as a whole just from one sample. But how does it compare to real-time PCR? I think people ask me this all the time. If we look at detection in real-time PCR, air is a little bit harder. The viral loads and things like that in air aren't always detectable and things like that. However, if we look at sequencing, almost 86 to 92% of our samples are actually positive for avian influenza. And that really brings up the question, is using sequencing actually more sensitive than qRT-PCR, and something that I think we need to discuss as a whole, as a collaboration. If we look at this kind of critically and we look at how we can actually detect full genomes compared to real-time PCR, you can see here quite clearly just from some spiked samples that even at a CT of 40.5, which would technically be negative for us based on gold standard diagnostics, we're still picking up a large amount of that genome at a almost 100x coverage across of it. So these are critical points that I think we need to look at in the near future. Because we really don't know what technically is the cutoff for real-time, for metagenomics and NGS in these samples, and something that we've been discussing for wastewater and things like that. But we really need to establish these standards as a whole to be able to use these as a gold standard diagnostic in the future. So what do we actually see? Here I've just made a heat map of detections in different kinds of samples in live bird markets. On the left, you can see air samples, and then I have different kinds of samples across the top. On the left here is actually viral families. So this is not individual virus species, it's viral families. So things like orthomyxovirus, gamma coronavirus, et cetera, et cetera. And then, of course, darker red means more reads for that particular family. And I think you can appreciate very clearly that air gives us a massive diversity of things that we're detecting. So we see a really massive diversity, even compared to, things like water overall. But we're doing the analysis now to understand what the differences are between these samples. But this pattern really holds true not only for live bird markets, but also for things like swine in abattoirs. Again, you see a really diverse detection in air. And then even in bat caves where we're detecting a large number of viruses in air compared to just feces, urine, and organs, which may be detected only partially of the time, or in individual samples or individual pools. The question then becomes, what do we do with this? Metagenomic data and sequencing of environmental samples really generates this cornucopia of data. It almost weighs us down, right? So that's what I've kind of used this middleman showing, where it's how do we make this heavy burden of data really reportable and actionable? And this is really what we've been focusing on now for the future. And so, we've been asking these questions, and they're ongoing right now, and I'd be happy to talk to you or work with any of you on this, is, what do we really do? And so we've been starting with sort of this red light, green light system that they use for AMR and seeing if that might work. So really focusing on what priority diseases are important for animal and human health in Cambodia. But we also need to validate these tests more, because obviously they're not our gold standards yet. They're novel. They're meant to be complementary, and they do not replace PCR at the current time. We're partnering with a number of stakeholders to understand this, both in country and globally, holding workshops and surveys to understand their needs and what the best way to do this is. We're really trying to provide new data visualizations, things like dashboards, and hopefully next time when I have more time, I'll be able to show you some of the interactive GIS and dashboards that we're making, that we can look at longitudinal active data in real time and look for increases in certain areas or over time that we can pinpoint. Also things like automating identification and alerts for stakeholders and for policymakers. And then, of course, ways to use the data differently. I think we need to really look at our surveillance and the way that we do things and start to use data differently. It may not matter that a certain pathogen is in a market or not at all times. What may matter is the diversity of pathogens within that market, how that correlates to things like climate change or urbanization, land use change, or even festival periods within the country, and then being able to monitor how interventions actually decrease that diversity of the virome or microbiome or whatever you want to call it, based on what we're actually doing, and really treating this as a pathogen agnostic approach. And so in the future, what we really want to do, obviously, is continue surveillance. We need to maintain this active longitudinal surveillance. We're doing further studies, and again, I'm happy to discuss and partner with anyone that's interested in moving forward with these things, especially public-private partnerships and really validating this technology in multiple settings. We're incorporating and testing even more novel technologies at all times. We have a number of studies ongoing that are looking at different kinds of air samplers, different kinds of drones, and different locations. And obviously understanding how these things incorporate for response and control, especially as people change their vaccination strategies, things like that are absolutely critical for the surveillance. And then, of course, we can't forget things like socioeconomics and behavioral studies that are absolutely critical for us to be able to use this data in a real way to make big changes. We cannot just survey pathogens and look at pathogens in the laboratory. We also need to understand knowledge, attitudes, and practices. We need to understand value chains. We need to understand drivers so that we can actually develop interventions that work to change the risk of these potentially risky, quote unquote, interfaces overall. And with that, I'll just say thank you. I can be contacted on a number of different ways. That QR code actually is my direct contact on WhatsApp and email if anyone needs to talk to me. And of course, I have to thank all of our partners that work with us over time. And I think I have a couple minutes for questions if needed. Thank you very much.
Thank you very much, Erik. A very nice presentation. So any question from the floor? Yes, please.
Thank you. My name is Junior. So I have a question about this collaborate environment, because you involve in your research so many solutions, some solutions based on AI, like some of them they are based robotics, and also some of them they are based on those kind of solutions in computer vision. So far, my question is, did you apply some kind of modeling so far so that we can have an open platform, even a private, but which can help to have a model, and based on that model, we can apply the kind of fine-tuning or transfer learning or data augmentation techniques in terms of gaining times and cost?
Right. Yes. So that's a very good question and a very good point. You know, this data is a very large database that we're continually adding to. I do have a couple of people that I work with at IPC in our GIS team, as well as our public health and epidemiology teams that are looking at some models. We are working with, and I don't know if anyone's in the audience from GeoSeek, but the Pasteur Network has just signed a collaboration agreement with GeoSeek, which is a very nice visualization and modeling platform. But it's my hope that we can actually provide this data set for people to work with us on modeling and understanding as well. So please do send me an email and we can chat more.
Okay, if I can add one more question. What can be the, if not now, maybe so far, what can be the main challenge of this kind of collaborate platform?
The main challenge with all of this right now is that, you know, from a research versus diagnostic perspective, we have to remember that most laboratories in many countries are reliant on gold standards, and many reference laboratories in the global north and other places are hesitant to adopt novel technologies until they are validated. So we need a lot more validation studies, and that's going to take some time. The other main issues, as I brought up, are really acceptability for action in real time. I think that's going to take some time, and hopefully we can move forward on that in the short term, but it's going to take medium to long term to really get that integrated into not only our diagnostic systems, but also our epidemiology systems as well as policy systems.
Okay, thank you very much, because the time is running out. Thank you very much, Erik. So hope to see you soon.
Yeah, thank you all. Yeah.